So good afternoon everyone. I hope each and every one of you is in the pink of health and happiness. I welcome you all to today's session. I am Nritya Koppalaramane and today we will be learning about cell, the unit of life. So the whole chapter is very much important. Each and every nook and corner there is been a question which have come in the previous year. So we will be learning, first we will be going through about the PYQs, then and there we will be learning about the concept according to the PYQs. Is it okay? So let's start the class. So cell, the unit of life. The first question which has come is omnis cellula e cellula. Omnis cellula e cellula. Who has given this concept? Regarding the cell division, it was first proposed by, it is Rudolf Virchow. What do you mean by omnis cellula e cellula? All cell arise from pre-existing cell. All cells arise from pre-existing cell. All cells arise from pre-existing cell, which means omnis cellula e cellula, which is given by, this is a cell theory, complete cell theory. So it was given by Rudolf Virchow. It is one of the important, very important question. The next question is cellular organelles with the membranes are. Cell organelles with the membranes. One of the cell organelle which does not have membrane is ribosome. So wherever the ribosome is, especially in the eukaryotic cell, one of the cell organelle which does not have membrane is ribosome. So wherever the ribosome is there, you just mark it as wrong. So we need to mark the cell organelles which have membranes. Yes, very good, very good. It is option A. Here ribosome is there. Here ribosome is there. Then here ribosome is there. So the option is C. Lysosomes have cell membrane, Golgi apparatus has cell membrane, mitochondria has cell membrane. The option A was for the previous answer, a previous question, I am sorry. Sri Lakshmi, is there any lag? Because we are already doing the second question. Okay, it was 1 minute before, sorry. Yes. So the option is option C. Lysosomes, Golgi apparatus and mitochondria has membrane, and these ribosomes doesn't have membrane. Now coming to the third question, which of the following structure is not found in the prokaryotic cell? Which of the structure is? Yes, nuclear envelope, very good. So we have ribosome which is in this thing, mesosomes are there, plasma membrane is there, but nuclear membrane, prokaryotic cell doesn't have nuclear membrane. Very good. Which of the following is not membrane bound? Again it is asked, which is not membrane bound? Which of the following is not membrane bound?
Ribosome.
Very good. Ribosome is not membrane bound. Membrane bound organelles are only present in eukaryotic cell. In eukaryotic cell, one organelle which doesn't have membrane is ribosome. Please remember this. Frequently they have asked question regarding ribosome. Frequently, it is ribosome is a membraneless organelle. Okay, doesn't have any kind of membrane. Now coming to the prokaryotic cell, the questions from the prokaryotic cell which is given in the second and third page in your textbook. Mesosome in the cell is, what is a mesosome? Do you know what mesosome is?
Special structure formed by extension.
Yes, special structure formed by extension of plasma membrane. So if this is a cell wall of the bacteria, and if this is a plasma membrane has like this special membrane bound extensions are there. Extension may be globular, may be tubular, may be lamella, anything. So these are called mesosomes. These are called mesosomes. What is the function of mesosome? Tell me. It helps in DNA replication, it helps in cell wall formation, it helps in DNA replication as well as distribution among the two cells when the cells are dividing. So these are the things this mesosome does. And also one of the other thing, especially what it does, energy synthesis. Energy, the ATP will be synthesized in that particular mesosome. Any doubts? ATP, because anyway the bacteria doesn't have mitochondria, so how do they produce ATPs, energy for the cell? That will be happening in the specialized structure. Those are called mesosomes. That is mesosome. So the answer to this question is the special structure, this one, formed by the extension of the plasma membrane. Given below are the two statements. Statement 1, in bacteria the mesosomes are formed by extension of the plasma membrane. Is it correct? Yes, the statement 1 is correct, as we have already studied about it. Statement 2, the mesosome in bacteria help in DNA replication and cell wall formation. Is it correct? Yes or no? Yes, it is also correct. So in bacteria mesosomes are formed by extension. Yes, mesosomes help in DNA replication, cell wall formation, both are true, which means statement A is correct and B is also correct. Option C is the correct answer. Statement 1, again statement based question. Mycoplasma can pass through less than 1 micron filter size. Is it correct? Because the mycoplasma is 0.1 micron meter. So can it pass through? Yes, it can definitely pass through. Mycoplasma are bacteria with cell wall. Do mycoplasma? Yes, mycoplasma doesn't have. So it is no cell wall. So this is wrong. Statement 2 is wrong. 1 is correct, 2 is wrong. So option D for Delhi is the correct answer. Which of the following statement about inclusion bodies? Inclusion bodies which are present in prokaryotic cell, which of the following is true? These are involved in ingestion of food particles. Is it true? For ingestion, like amoeba, is it food vacuole? No, it is not. So A is wrong. They lie freely in the cytoplasm. So we need to find the incorrect one. Incorrect. Yes. Yes, they lie freely in the cytoplasm. Yes. These represent reserve food materials in the cytoplasm. Yes. They are not bound by any membrane, as it is seen in the prokaryotes. It doesn't bound by any kind of membrane. This is also correct. So we need to find the incorrect one. That is option A is a correct answer. Inclusion bodies, again inclusion bodies are very important. Again and again they have asked question. Inclusion bodies of blue green, purple and green synthetic bacteria have what? They have something called gas vacuoles, which helps in maintaining the buoyancy in aquatic environment. So these have gas vacuoles where the gases will be filled inside the cytoplasm, inside this. But as you can see vacuoles here, don't think it is membrane bound. It is not membrane bound. If you see the vacuoles, like same in the eukaryotics, how we see the vacuoles, you might think that it is membrane bound. No, gas vacuoles are not membrane bound. Inclusion bodies are also not membrane bound. Anything inside the prokaryotics is not membrane bound. Everything is membraneless. Okay. Only eukaryotes have membrane bound organelles. Prokaryotic doesn't have any kind of membrane in the cell organelles. Understood? So it is not membrane bound, which means there is no membrane present in the gas vacuoles. The size of the PPLO, that is just what? PPLO is a pleuropneumonia like organism. PPLO is a mycoplasma, right? Mycoplasma.
Option C.
Option C. Just now we discussed. It is 0.1 micrometer. That is here. That is option C. Which one of the following is not a prokaryote? So Saccharomyces, which means this is a yeast. Yeast is a fungi. This is a eukaryote. Mycobacterium, prokaryote. Nostoc is a prokaryote. It's a blue-green algae. Oscillatoria is also blue-green algae. All these are prokaryotes. You need to find not a prokaryote, that is Saccharomyces cerevisiae, which is a yeast, is a eukaryote. Many ribosomes may associate with a single mRNA. So in prokaryotes, what happens? From the DNA, mRNA will be synthesized. This mRNA, see, everything is going on in the cytoplasm itself, right? Cytoplasm. So if this is a mRNA, we can see the ribosome, many ribosome attached to single mRNA. mRNA is one, but the ribosome is many. It will be attached like this. So all the ribosome will be doing their own protein synthesis here. The protein synthesis will be taking place here. Polypeptide chain ka synthesis will be taking place. Okay. So like this. This particular structure here with the ribosome and mRNA, this particular structure we call it as polyribosome. Otherwise it is also called as polysome. This kind of structure, in one single mRNA there will be multiple ribosome will be attached and they will be synthesizing the protein. Such thing we call it as polyribosome or else polysome. That is what they have asked. Single mRNA to form multiple copies of polypeptide simultaneously. Such strings of ribosome are termed as option A, that is polysome. Understood? Option A, that is polysome. Very good. Which of the following components provides sticky character to the bacterial cell? For adherence.
Glycolate.
Yes. If this is a cell, there will be the outer this thing, that is glycocalyx. Very good. What it does actually? What there are two types, right, for the glycocalyx. Just tell me what are those. Glycocalyx can be loosely bound, that is called loosely bound, that is called slime layer. Or thick it is called capsule. And this capsule, can you see this capsule actually gives the virulence character. Virulent or such the bacteria which have the capsule layer out, they have the virulent character. Just a quick plus point in that. 14. Select the wrong statement. Can you select the wrong statement? Cyanobacteria lack flagellated cells. Yes, they don't have flagella. Pili and Fimbria are mainly involved in motility of the bacterial cell. Yes, this is wrong. Why? Tell me. What Pili and Fimbria does? What pili does actually? Pili helps in conjugation, okay, which means transfer of DNA from one to another, okay, or sharing we can call it as, that is conjugation. And what fimbriae does? Adherence, like sticking onto the substrate. If they want to stick onto substrate something, if this is a substrate and if they have to stick onto it, it help with the help of fimbriae, they adhere to the substrate. That what it does. Pili and fimbriae is not helping in motility or locomotion. Bacterial cell wall is made up of peptidoglycan, correct. Mycoplasma is a wall-less microorganism, correct. This is D4L is a correct answer. Select the mismatch. Protists are eukaryotes, yes. Methanogens are prokaryotes. In prokaryotes, what are they? Tell me. Archaebacteria, eubacteria. Methanogens are archaebacteria. Yes. Methanogens are archaebacteria, eubacteria. Just tell me, students. Just type it.
Archaebacteria.
Yes, very good. Archaebacteria. Gas vacuoles, green bacteria, correct. Large central vacuole, animal cell. Where the large central vacuole will be? In the plant cell. So, which makes D4L as a correct answer. A complex of ribosomes attached to single strand of RNA is known as, again they have asked, just now we discussed. Tell me, what is the answer?
Polysome.
Yes, very good. Dhanashri, very good Dhanashri. It is called polysome, otherwise it is also called, what it is called? Yes, polyribosome. Very good, students. Which of the following is not an inclusion body found in prokaryotes? Tell me. Inclusion bodies are glycogen granule, phosphate granule, cyanophysin granule. Glycogen granule, where they produce, where they store glycogen, that is food. This is storage. Okay. Phosphate granule, the phosphate will be stored, again storage. Cyanophysin granule, pigment will be stored. Anyway, all storage. What about the polysome? Polysome or the polyribosome is a structure we can see that is not an inclusion body. It is just mRNA. On top of the mRNA, it is the ribosomes are attached here and there. That is called polysome. So, which is not an inclusion body? Polysome is not an inclusion body. Chromatophores take part in. What are chromatophores? In say mesosomes, if this is a bacterial cell, if we see the mesosomes are there, right? Mesosomes. Okay, especially in photosynthetic bacteria, if they have pigment present in this pigment present, these are called chromatophores. And what they does, pigment present, which means they do the photosynthesis, because as they are prokaryotes, they doesn't have chloroplast, membrane bound. Chloroplast is membrane bound. They don't have chloroplast. They have same mesosome which will be helping in photosynthesis, the mesosome which will be helping in respiration. So, the same mesosome which has the pigments present, we call it as chromatophores. What is the function of it is? The function is photosynthesis. That is D4L. Understood? A quick thumbs up please, so that I get to know that you have understood. Thank you so much. The structure that help in some bacteria to attach to rocks and to host tissue are Mesosomes, no, the respiration photosynthesis. Hold fast, where hold fast is? In the brown algae, if you can see. Rhizoids in the bryophytes. So it is Fimbriae. Such a easy question. The motile bacteria are able to move by. Is the pili helping in moving? No. Fimbriae? No. Cilia? No. Flagella? Yes. You might ask, like, why cilia is not helping in moving? So here they have asked about the bacteria. Cilia you can find in eukaryotes. Eukaryotes. Example, paramecium you can see. In our body also you can see in the respiratory tract you can find cilia. So especially it is exclusive for eukaryotes. You can find only in eukaryotes. Flagella you find in prokaryotes as well as eukaryote you find, but cilia is for only eukaryotes. So remember this, don't forget this particular point. It is not cilia, it is only flagella. 21. Statement 1. Concentrically arranged cisternae of Golgi complex. So now we have come to endomembrane system. That we will be discussing only about the Golgi complex, lysosomes, then vacuoles and this endoplasmic reticulum. Statement 1. Concentrically arranged cisternae of Golgi complex are arranged near the nucleus with a distinct convex cis or maturing and concave trans or forming phase. Is it correct? Correct or wrong? Unmute yourself and answer the question. Because yes, it is incorrect. Why? Then if this is a cell, and if this is a nucleus, you can find the Golgi like this. Okay. So this particular phase is cis phase. This particular phase is proximal phase. Here they have mentioned distinct. No, not distinct phase. This is wrong. Cis and maturing phase. Maturing is also wrong. So this is the forming phase. Forming phase. Okay. This is cis phase and also convex phase. Otherwise it is called convex phase. So this particular phase here, this is cis phase, proximal phase, or else we call it as forming phase, otherwise convex phase. This particular, which is towards the plasma membrane, this is plasma membrane. Plasma membrane. Towards the plasma membrane, this is called concave phase. Concave. It is called distal phase. Away, distance, towards the distance, other distance. Distal phase. Then it is called as maturing phase. Maturing phase. Otherwise it is also called as trans phase. Trans phase. So please remember, towards the nucleus it is called cis phase, proximal phase, forming phase and convex phase. Towards the plasma membrane it is called concave phase, distal phase, mature phase and trans phase. So which makes this as a wrong answer. Statement 2. The number of proteins are modified in the cisternae of the Golgi complex before they are released from the cis phase. Is the releasing taking place in the cis phase? The release taking place in the trans phase. The number of proteins are modified in the cisternae of the Golgi complex. Yes, that is correct. Before they release from the trans phase. So both statement 1, statement 2 are wrong, which makes statement 1, statement 2 are false. A as a correct answer. Any doubts? Any doubts? Yes, we are moving on to the next question, that is, which of the following are not considered as the part of endomembrane system? Endomembrane system has endoplasmic reticulum, Golgi complex, then lysosomes and vacuoles. Other than these four, rest of are not endomembrane system. Is the mitochondria not endomembrane system? No. Endoplasmic reticulum? Right. Chloroplast? No. Golgi complex? Yes. Peroxisomes? No. So we need to find which are not. So it is A, C and E. A, C, E are not the part of endomembrane system. That is, C is the correct answer. Which of the following statements are correct with respect to Golgi apparatus? We need to find which are the statements are correct with respect to Golgi apparatus. It is the important site of formation of glycoprotein and glycolipids. Yes, it is one of the main function. Yes, it is the main function of Golgi apparatus. It produces cellular energy in the form of ATP. No. Who does this? Mitochondria does this. It modifies the protein synthesized by the ribosomes on the ER. Yes, it modifies the protein, definitely. It packages, transports everything, so it modifies. It facilitates the transport of ions. No, who does this? Plasma membrane does this. Facilitate the transport of ions. The membrane, any membrane can do this, not only plasma membrane, tonoplast also does. The membrane does that. It provides the mechanical support. Who does the mechanical support? Is provided by cytoskeleton.
Cell wall.
Cell wall, yes. Cell wall, cytoskeleton, yes. So not this. It is only A and C as right answer. That is B for Bombay. B for Bombay. A and C is the right answer. If the pH of the lysosome, what is the pH of the lysosome? It is acidic always. Acidic is increased to alkaline. Okay, we are increasing the pH to alkaline. What will be the outcome? So lysosome enzymes are active in the acidic pH. So if we increase the pH to basic, that is more than 7, if we increase that, what happens? Here they are active, in the basic pH they will become inactive. So more active? No. Hydrolytic enzyme will function more efficiently? Never. And hydrolytic enzyme will become inactive? Yes, they become inactive. The lysosome enzyme will be released into cytoplasm? No, they need particular signal for that. So it is C as the right answer. Any doubts regarding till now, students? Till now?
No, ma'am.
Yes. Thank you so much. The cell organelles that are included in the endomembrane system are, again they have asked the same question. Okay. So here ER, Golgi complex, lysosomes and vacuoles. We have got the answer in the first this thing itself. A as a right answer. Here mitochondria is not the endomembrane system. Mitochondria again, not here mitochondria again. So it is A is the right answer. With the first reading the first option, we got to know that A is the right answer, because endomembrane system consists of endoplasmic reticulum, Golgi complex, lysosome and vacuoles. So the questions are very easy from this particular chapter, but there are more questions they have asked. It becomes very important chapter. Which of the important site of formation of glycoproteins and glycolipids in eukaryotic cell? I said right. So proteins are formed in ER, and formation of glyco, which means adding the sugar or carbohydrate, you can call it as any carbohydrate, adding to it, where it happens. So lipid will be synthesized in smooth endoplasmic reticulum, proteins will be synthesized in rough endoplasmic reticulum, but glycolating it, this happens in Golgi bodies in the yes, one of the very important function of Golgi bodies are glycolating it, that is adding the sugar to the produced proteins and lipids. Proteins are synthesized in rough endoplasmic reticulum, lipids in the smooth endoplasmic reticulum. Glyco, that is adding the sugar, takes place in the this one, Golgi complex. Which of the following statement is not correct? We need to find which is the non-correct one. Lysosomes have numerous hydrolytic enzymes. Just tell me what are the hydrolytic enzymes which these lysosomes have. They have nuclease, amylase, lipase, protease. So this is wherever it comes, right? These enzymes, actually what they do, it degrade the protein. It degrade the protein. Nuclease, nucleic acid, it degrade the nucleic acid, and amylase, carbohydrates, any kind of starch, maybe glycogen, any kind of carbo, and lipase degrade the lipid. Okay, so these are the hydrolytic enzymes, main hydrolytic enzymes which will be there in the lysosomes. Okay, so this makes this statement correct. The hydrolytic enzymes of the lysosomes are active under acidic pH. Yes, all these will be active in the acidic pH. Okay, and lysosomes are membrane bound structures. Eukaryotic, yes, they definitely are the membrane bound structure. Lysosomes are formed by the process of packaging in the endoplasmic reticulum. Is in the endoplasmic reticulum? Tell me.
Golgi complex.
Golgi bodies or Golgi complex. Golgi complex. So which means D for Delhi as a right answer. The Golgi complex participate in what it participates in? Fatty acid breakdown. Formation of secretory vesicles. Yes. Where fatty acid breakdown takes place, just tell me. Fatty acid breakdown. It takes place in mitochondria. Takes place in mitochondria. Respiration in bacteria, mesosomes. Activation of amino acid, cytoplasm. Formation of secretory vesicles will be formed by the Golgi complex. B for Bombay is the right answer. Which of the following events does not occur? Does not occur in the rough endoplasmic reticulum. Protein foldings, phospholipid synthesis. Yes, the protein folding, yes, it does. Protein glycosylation, yes. Cleavage of, yes, whatever it does with the protein, yes, it does. And phospholipid synthesis, where it happens, you just tell me. Lipid synthesis. The lipid synthesis will be taking place in the smooth endoplasmic reticulum. D for Delhi as a right answer. The cell organelle containing hydrolytic enzyme is, just now we discussed about it. Very easy. Everybody knows that. Yes, it is the lysosomes which have hydrolytic enzymes such as nuclease, protease, amylase, lipase, such as that. Select the correct matching in the following pairs. Rough endoplasmic reticulum, synthesize glycogen, yes. Oxidation of fatty acid, no. Smooth endoplasmic reticulum, synthesis of lipids. The previous D for Delhi, the previous question we did that. And just tell me, very good. Just tell me, what are the other functions of smooth endoplasmic reticulum? What are the other functions of smooth endoplasmic reticulum? It synthesizes lipids and sterols. Yes. The osmotic expansion of cell kept in water is chiefly regulated by vacuole. Very good. And how it does that? So for example, yes, tell me. So for example, this is the plant cell. Okay. So this is a large vacuole, is this central vacuole, large vacuole. So this vacuole will be having high solute concentration. Solute concentration will be very high. Okay, as solute concentration is very high, this is kept in water. For example, say this is kept in water. This is kept in water. As there is high solute concentration, what happens? The water from here get into the cell. For osmotic balance, as there is solute concentration, or this water get into the vacuole. This is vacuole. As the water get into it, it enlarges. It enlarges. And it put pressure to the outer wall. That's why the cell swells up. It put pressure, osmotic pressure. That is, we call it as Turgor pressure. Pressure on the cell wall. As the plant cell has a cell wall, anyway it doesn't burst. Plant cell doesn't burst. It has cell wall here, but it put the Turgor pressure on the cell wall. That's why there is a this thing like very rigid kind of structure it forms. That is vacuole, because vacuole has more number of solute concentration, and because of that the water get inside, and it inside that it swells up, and it put the pressure on the walls of the cell wall of the plant. Which of the following organelle is in the figure correctly matches with its function? So what is this? Here if you see, and yes, also we could see the ribosomes on top of it, which makes this as a rough endoplasmic reticulum, isn't it? It makes as a rough endoplasmic reticulum. Just tell me, what is the function? What is this? This is rough endoplasmic reticulum. And what is the function of rough endoplasmic reticulum? The rough endoplasmic reticulum synthesize protein. Not, I mean, this definitely not synthesize protein. It forms the site for synthesis of protein, right? Size, which synthesize protein? We know that ribosome does that, but rough endoplasmic reticulum actually makes the site, which means it give the place for, अरे आ जाओ राइबोसोम, यहाँ मेरे ऊपर बैठो और प्रोटीन सिंथेसिस करो, ऐसे वाला site, place ribosome gives, place rough endoplasmic reticulum gives. Yes. So which means rough endoplasmic reticulum, and the function of it is protein synthesis. A is the right answer. A major site for protein synthesis of lipid. Easy. It is lipid synthesis takes place in smooth endoplasmic reticulum. That is, C is the right answer. The Golgi complex plays a major role in, what is the
Option A.
for post translational modification of protein and glycosidation of lipids? Yes, option A. Why other option are wrong? In trapping the light and transforming it in the chemical energy, who does this?
Mitochondria.
Trapping the light energy and transforming it to chemical energy. Mitochondria, when mitochondria started to trap energy, then we would be like standing in the under the sun and photosynthesizing, right? Because we have mitochondria. Mitochondria is not going to trap light energy. Then there is no food crisis in India. LPG का कोई समस्या ही नहीं होता ना. Isn't it? We would go under 1 hour, we'll be under the sun, we would get energy, then come back. वो जादू बन जाते हम यार, जादू जादू वाला. So it is chloroplast does this. In digesting proteins and carbohydrates, digestion, lysis, who does this? Lysosomes. Then energy transferring organelles. Now energy synthesizing we can say, transferring, that is fine, mitochondria would do that. Yes. So 36. Now we came to mitochondria, plastids and ribosomes, part of the chapter. The DNA present in chloroplasts. As chloroplast has its own DNA, chloroplast has its own ribosomes. So the DNA can be converted into mRNA, and mRNA with the help of ribosome will be converted into proteins. So chloroplast has all these things. It has its own DNA, it has its own mRNA, ribosome, proteins. Also, it is dependent on other proteins, okay, for synthesis. It also depends on cytoplasm, that is nucleus it is dependent on. So we call it as semi-autonomous. It is not completely autonomous, but semi-autonomous. Why? Because it has its own DNA, mRNA, ribosome, proteins, it has its own, and it is also dependent on nucleus for some other. So it is called semi-autonomous. What type of DNA it is present in chloroplast? It is a circular DNA as well as double-stranded DNA it has, and it is a circular DNA. So it is D4 Delhi is a correct answer. Understood? D4 Delhi is a correct answer. It's a circular DNA. It is not the linear DNA as the nuclear DNA. Nuclear DNA is linear DNA and nuclear DNA is linear. It is a circular DNA and it is double stranded. Read the statements. Mitochondria and chloroplast are both double membrane bound organelles. Is it correct? Yes, both have inner membrane and outer membrane, isn't it? We have inner mitochondrial membrane, outer mitochondrial membrane, inner chloroplast membrane, outer chloroplast membrane. So this makes correct. The inner membrane of the mitochondria is relatively less permeable as compared to the chloroplast. Tell me, is it true or false? Tricky statement. So we know. Okay, let's begin from the beginning. Okay, if this is a, what is this? Tell me. This is the mitochondria. Outer mitochondrial membrane, inner mitochondrial membrane. Okay, so outer mitochondrial membrane is more permeable. Okay, more permeable. It allow anything inside. Okay, fine. I mean, not anything, but it is not that selectively permeable. It is more permeable to many of the things, maybe it any molecules or maybe it ions, anything it can leave. But inner mitochondrial membrane is selectively permeable. Is selectively permeable. Okay. For example, like many people come into your house, right? Those who know, you can let them inside your house, but into your room not everybody can come, right? Only you can go or your family members can go, such kind. So outer, the first door, I mean the main door, many are allowed, like relatives are allowed, friends are allowed, these that many are allowed, but into your room only you are allowed, your family members are allowed, such very selectively permeable, right? So the inner one is like your room and it is selectively permeable. The outer membrane is more permeable. In the same case for the chloroplast also. Chloroplast has two membrane. The outer membrane is more permeable. Sorry. Outer membrane of the chloroplast is more permeable and inner membrane comparatively less permeable or selectively permeable. Okay. Now both inner membranes are selectively permeable, but when compared to the mitochondrial membrane inner membrane, mitochondrial inner membrane and chloroplast inner membrane, which is more permeable, or like which has a more selectively permeable? That is the question they have asked when compared to inner mitochondrial membrane and inner membrane of the chloroplast, which is more. So, inner membrane of the mitochondria is relatively less permeable? No. Actually, when compared to the inner membrane of the mitochondria, inner membrane of the chloroplast is more selectively, which means inner membrane of the mitochondria is relatively more permeable than the chloroplast, which means now the inner membrane of the mitochondria is your room and you have the cupboard, right? Now the cupboard is very less permeable. Even if family members also cannot open the cupboard, your cupboard, kind of. That cupboard is the inner membrane of the chloroplast. That is even more selectively permeable. Understood? So, which means the inner membrane of the mitochondria is relatively more permeable, kind of more permeable, because it is room as compared to the chloroplast. So, now they have given less, which means this is wrong. Understood? Any doubts? Any doubts, or should I explain it again? साक्षी, डाउट है या नहीं है?
No, ma'am.
अच्छा, नहीं है डाउट. You understood, which means?
Yes, ma'am.
Thank you so much. So, inner membrane of the mitochondria is relatively more permeable than the chloroplast is a correct statement, but here it is given less, so it becomes wrong. So, 1 is correct and 2 is wrong, which means A is a correct answer. Which of the following pair of organelles does not contain DNA? Mitochondria? Do mitochondria has its own DNA? Yes, mitochondria has its own DNA, but lysosome doesn't have. Chloroplast has, vacuole doesn't have. Lysosome also doesn't have, vacuoles also doesn't have. Nuclear envelope?
Option C.
Oh, does not contain, yes. So, it doesn't have, it has. So, which means A. C. Option C is the correct answer. This both doesn't have. This whole pair doesn't have the DNA. So, in this in this pair, one each has the DNA. Which of the following statement regarding the mitochondria is incorrect? We need to find the incorrect ones. Outer membrane is permeable to monomers of carbohydrates, fats and proteins. Outer membrane, I said permeable, yes. Enzymes of electron transport chain are embedded in the outer membrane. Electron transport chain कहाँ होता है? Inner mitochondrial membrane में. Inner membrane is convoluted with the infoldings. Yes. What do we call it as? We call it as cristae. We call it as cristae. Mitochondrial matrix contains single circular DNA. Yes, it is single circular DNA, circular double stranded DNA molecule and ribosome. Yes, it does have. So, this is also correct, which means B for Bombay is the wrong answer. Why? B for Bombay is the right answer. This statement is wrong. Why? Because it is inner mitochondrial membrane has this ETC ka enzymes. Which of the following cell organelle is responsible for extracting energy from carbohydrates to form ATP? Easy, very easy. It's a cakewalk for you. Which one is that? Mitochondria. D for Delhi. Very easy. So, if you can see this, in this particular chapter, there are so many, many questions comes, okay, around 3 to 4 questions comes, but those questions are very easy, right? Are you feeling any difficulty in solving such these questions? Very easy. So, once if you go through the NCERT, it will be very easy for you to answer the questions. So, you can score marks. So, you can score maximum marks from this particular chapter. Mitochondria and chloroplast are semi-autonomous organelles. Now, just we said about because it has its own DNA, proteins, RNA, everything. So, they are semi-autonomous organelles. Formed by division of pre-existing organelle, semi-autonomous, yes, and they contain DNA but lack protein synthesizing machinery. Is it correct? Protein synthesizing
No.
they have ribosomes. They have ribosomes. They have mRNA. इसके अलावा क्या चाहिए? They have it, right? So B is wrong. A is right. A is true, B is false. That is, C is the right answer. The structure that are formed by stacking of organized flattened membranous sacs in the chloroplast. I will just draw the chloroplast diagram, okay. So, if this is the chloroplast, outer membrane, yes, grana is the correct answer, very good. Inner membrane, we have thylakoids. So, these are the organized flattened membranous sacs they have called, those are what they have mentioned here, those are the thylakoids. So, there will be thylakoid stacking, as I previously said, like a coins which you stack one on the other. So, this is the stacking, and these are also connected. So, one individual unit we call it as thylakoid, and all these things we call it as grana, and this particular thing we call it as stoma lamellae, and this is called stoma, and this is outer membrane, this is inner membrane. With that, it has its own circular DNA which is double stranded. Also, it has its own ribosomes. Also, there will be storage, starch granules also present. Starch granules. So, this is a chloroplast. Okay. So, which makes D for Delhi as a right answer. DNA is not present in, DNA वे कहाँ present नहीं है, न्यूक्लियस में ऑब्वियसली DNA प्रेजेंट है, माइटोकॉन्ड्रिया में प्रेजेंट है, इसमें भी प्रेजेंट है, राइबोसोम में DNA नॉट प्रेजेंट. सो वेरी इजी. In which structures perform the function of mitochondria in bacteria? Bacteria में respiration, yes. So after we solved so many questions, you might have already known, right, how important the mesosome is. Mesosome wala part, how many times they have asked? Repeatedly they have asked. Which of the following function is carried out by the okay, now we have come to cytoskeleton, cilia and flagella, centrosome and centrioles. Okay, that part of the lesson. Which of the following function is carried out by cytoskeleton in the cell?
Transportation.
नहीं मेरा बच्चा, ऐसा नहीं है. Transportation, no. So here, see, this will be motility. So what do you mean by cytoskeleton? You tell me. I'll tell you. If have you seen this sleep well mattress ka ad? Sleep well mattresses. So mattresses, this cushion wala this thing will be there, no? So if this is that mattresses, if this is that bed, so if somebody sits on it, after they get up, it will regain its structure, right? Because it has spring inside. Do you know? So if somebody sits on it, it will form a dent like this. Okay. After they get up from that, Yes, yes.
Regain their position.
Huh?
Regain.
Yeah, sure. Yes, it regains its position. So why? Because there is spring attached to it. Springs. So in a same, ये स्प्रिंग वाला जो फंक्शन स्प्रिंग कर रहा है ना, वैसे ही इस फंक्शन ना साइटोस्केलेटन भी करता है इनसाइड द सेल. आई विल टेल यू. If this is a cell, if other cell is there present, if this is another cell, so this cell can deform itself. ऐसे अंदर तक जाता है. If when you remove this particular cell, formal cell will definitely regain its position back. How does it regain its position back? क्या कर रहा है? Inside ना, there is actually this cytoskeletons will be present all everywhere. So if you see, we have already learned like cell है, cell wall. I mean in animal cells, if we see cell membrane, nucleus is there, organelles is there, that is there, this is there. Then we go to cytoplasm is there. So actually, what is holding its particular this thing? Holding the structure of all these things, it is the cytoskeleton which is there inside the cytoplasm, which will be holding its structure. Okay, so that will be, so that is the cytoskeleton. You can give the spring as a example. This Kurlan ka spring है ना, that will be cytoskeleton. Understood? D for Delhi as a right answer. Match the following columns. Smooth endoplasmic reticulum, lipid synthesis. Rough endoplasmic reticulum, protein synthesis. Golgi complex, glycosylation, and centriole, spindle fiber. A to B1. It is D for Delhi as a right answer. Microtubules are the constituents of. Microtubules are in cytoskeleton, cilia, flagella, centriole. So here it is not in peroxisome, it is not in the chromatin, it is not in the nucleosome. Microtubules are present in spindle fibers. ये centriole आगे बनाता है ना spindle fiber, it makes spindle fiber. In animal cell, when the cell is dividing, यहाँ the structure, if you see, these are the structure, if you see, these are centrioles, and it will be holding the kinetochore as a spindle fibers. So this is spindle fiber. Spindle fiber is also made up of the microtubules. Centriole is also made up of microtubules. Cytoskeleton, cilia, flagella, everything. Okay, so it is B for Bombay as a right answer. The solid liner endocytoskeleton elements having a diameter of 6 nanometer are made up of single type of monomer are known as. So there are several types of this thing cytoskeleton mein tubules. One is microtubules, intermediate filaments, and microfilaments. Microfilaments. So this microtubules are actually larger. Okay, they are around 25 nanometer width. Okay, diameter is 25 nanometer. Intermediate, as they are very intermediate to this, it will be having around 12 nanometer width. Microfilaments will be having 6 nanometer. They are very small. Okay, these are having microfilaments having 6 nanometer. They are very small. So which means here it is a diameter of 6 nanometer is actually microfilaments. And lamins is one of the example for this intermediate filaments. Lamins. This lamins actually they will be forming the nuclear lamella. Nuclear lamella. They will be forming nuclear lamella. Understood? So according to this question, this is actually 2014 question. Now it's not in the syllabus. So still, as the question we are discussing about, the answer to this question is microfilaments. There are types of cytoskeleton filaments present. One is microfilament, microtubules, and intermediate filament. Intermediate, by the name you can see it is intermediate to microtubules and microfilaments. Microfilaments actually comprise more. These are very much compared to other two. Understood? Match the following. Centriole. What is a centriole? Centriole, yeah. Basal body for cilia and flagella. Cilia and flagella. What is the arrangement of cilia and flagella? It is 9+2 arrangement, right? 9 doublet and 2 singlet. Actually the centriole, what is the arrangement? It is a cartwheel arrangement. It is 9+0. What are 9? 9 are triplets. But the basal body of the cilia and flagella, the cilia and flagella, the filament is the 9+2. The basal body is the centriole, that is 9+0 is the arrangement. Screen is too white. I'm sorry. Just was it for the previous also? Oh yeah. Oh, thanks. Nuclear lamella means the membrane, nuclear membrane. Lamella जो भी हो बेटा, lamella मतलब होता है membrane, plasmolemma, lamella. Now it is okay? Not clear, ma'am? Now it's fine. Yes. Yes, thank you so much for letting me know. So the smooth endoplasmic reticulum, oh okay, fine, this is done. We were here. So this is about central basal body of the cilia and flagella. Chlorophyll present in thylakoids. Cristae is the infolding of the mitochondria. Ribozyme. Please tell me what are ribozyme? Ribozyme is a RNA. Ribozyme is a RNA which act as enzymes. RNA enzymes, ribozymes. RNA which are enzymes are called ribozymes. We say that this one, enzymes are protein in nature. Not all enzymes are protein. There are certain enzymes which are RNA also, that we call it as ribozyme. Okay, ribozyme. Ribozyme which is a RNA which is a nucleic acid. That's why we marked it to nucleic acid. Okay, so it is A as 4, B2, A4, B2, B for Bombay as a right answer, C1D3. Yes. Any doubts till now? Any doubts? So now coming to the nucleus and microbodies, last part of the lesson. Okay, last part of the lesson. We are going to match it. Fleming. Fleming, actually he is the one who discovered or gave this, we can call it as coined chromatin. Okay, he stained it. When he stained it, he could see that that particular stain was taken up by the DNA, that is chromatin, and he could see it under the microscope as it was chroma means color. It could take up the color and he could see under the microscope. So he named it as chromatin. It is Fleming who did that. Robert Brown, we know that. Robert Brown first discovered the nucleus. George Palade. Palade particles are the ribosomes. Yes, Palade particles are, ribosomes are also called Palade particles. Camillo Golgi, dish-shaped sacs or the cisternae near the cell or the nucleus, that is Golgi apparatus, which gives the answer A2, B4, A4, A is the answer, C3 and D1 is correct. Any doubts? Match the following again. Match the following. Metacentric, we know based on the centromere position, we are dividing, segregating into 4 types, that is metacentric, submetacentric, agrocentric and telocentric. Metacentric means it is right in the middle of the 2 chromatid. So if this is the centromere, it is in the middle. So P arm, this is P arm, this is Q arm. P is equal to Q. The length of the P is equal to Q is metacentric. Submetacentric means slightly away. This one, slightly away. If this is a slightly away, this is the centromere, slightly. So P arm is less, Q is more. This is Q arm is more than P arm. Acrocentric, centromere situated close to its end. Please remember, it is close to its end. When it is given close to its end, forming one extremely short and one extremely very long, that is acrocentric, not the telocentric. Telocentric comes terminally. When it has the word terminally, then only mark it as telocentric, otherwise it is acrocentric. Which one is acrocentric? This one. Q arm is very long, okay, very long than P. This is P, this is Q. Q is very long, okay. Here it is, the centromere is towards the end, extremely short one end and extremely long another one. Now coming to the telocentric. Telocentric is, this is the centromere. and this is the Q arm. You cannot see the P arm, you cannot even see the P arm. So which is Q is very much large than P. So that is telocentric. Towards the telomere, this position of this chromosome, this position we call it as telomere. Telomere. If the centromere is present towards very last tak, then it is called telocentric. So now A to B3, C4 and D1. D4 Delhi as a right answer. Any doubts? Oh, this is still not visible correctly. Wait 1 second. This part is not visible correctly, right? No, fine. No, fine. No, it's fine.
Want to change?
No. So yes, it's 15 minutes left. We'll cover it up first. So now, so previously everything clear, right? This part was not clear previously, right? So yes, we move on. We are going to match the list, that is F1 particles. F1 particles you can see, that is F0, F1 particles in the ATP synthase, that is in the mitochondria. Histones you can see in chromosome, that is if this is a DNA is wrapped around the histone. This is the histone wrapped around in the histone. Here also. Yes. So now axonym you call it in that if you cut open the flagella, you see that these things, right? The doublet and the singlet here. This whole thing is called axonym. You can see it in cilia or flagella if you cut open the cilia or flagella. Okay, cisternae you can see it in the Golgi apparatus. If this is the Golgi apparatus, this thing we call it as cisternae. So A4 B1, C2, A4 B1. Oh, C2, D3. Yes, sorry, this one. B for Bombay is the correct answer. Choose a correct answer. Nucleolus. Nucleolus is a site for active ribosomal RNA synthesis. If you see the nucleolus, it synthesizes rRNA. This rRNA will be ultimately synthesizing the ribosome. That's why we call it as ribosomal RNA. Centriole is a organization like cartwheel. Leucoplast. In chloroplast there are three types: chloroplast, chromoplast and leucoplast. Chloroplast green pigments will be there. Chromoplast, other than green pigments like yellow, orange, red, these pigments will be there in the chromoplast. And leucoplast, actually it stores the food, such as a uroplast store protein, a lyoplast store oil, and a myeloplast store starch. Okay, so that is storing, leucoplast actually stores the nutrients. Golgi apparatus, site for formation of glycolipid and glycoprotein. A3Bu2, this one, C as a right answer, C4D1. Match the list 1 and 2. List 1, axonym is a cilia or flagella, discussed. Cartwheel pattern is a centriole. Cristae is the infolding of mitochondria. Satellite, it is a satellite, actually it is present on the chromosome. Okay, I will tell you. If this is a chromosome, this is the centromere. Centromere is a primary constriction. Okay, there will be sometimes there will be secondary constriction here, this, okay, secondary constriction here, this particular thing is called secondary constriction. This part above the secondary constriction, if you see this one above the secondary constriction, this one, this is called SAT DNA or satellite DNA. These are non-stainable. You cannot stain it. Okay, chromosome is a stainable, right? We can stain it, but when it comes to satellite, it is non-staining part. Why? Because it is so small, you cannot even see that. Okay, how do you stain it? How that can be visible? Understood? It is such a very small. अगर मैंने very छोटा सा एक चीज़ देके अगर तुम्हें बोलूँ कि paint करो, you put the color on it, can you able to do it? No. That's why it is non-staining. Stain नहीं. Okay, no, there will be no stain in that. That is non-staining. That is satellite. So it is A2B1. B for Bombay as a right answer. Again, match the following. So many match the following questions are there. See, it's the same thing which we did in the previous year, I think. Again, same questions they have asked about the metacentric, submetacentric. So here, metacentric is a centromere in the middle. Acrocentric is situated to the end, yes. Submetacentric, slightly away, yes. And telocentric at the terminal end. वो ऑप्शंस भी चेंज नहीं किए, सिर्फ द सबमेटासेंट्रिक को सी में डाल दिया और एक्टोसेंट्रिक को बिल में डाल दिया, इतना ही चेंज किया उन्होंने और कुछ चेंज नहीं किया. सो इट इज़ लाइक रिपीटेडली दे आर आस्किंग इट. A3B1. Okay, B for Bombay is the right answer. Which of the following is incorrect? Incorrect statement. Microbodies are present both in plants and animals. Microbodies are present in both plants and animals. This is correct. The perinuclear space forms a barrier between the materials present inside the nucleus and that of the cytoplasm. Sure, this is correct. Nuclear pores act as a passage for proteins and mRNA molecules in both the direction between nucleus and cytoplasm. Correct. The mature sieve tube elements possess the conspicuous nucleus. No, nucleus is absent here. In the mature sieve tube, you don't find the nucleus. That's why the sieve tubes have the companion cells, right? Companion cells. This companion cell has nucleus. And these companion cells are the one which will be helping sieve elements. These are the companions are the one which will be regulating the sieve elements, because sieve they themselves doesn't have any kind of nucleus, especially the mature sieve doesn't have, okay, as a nucleus. This is wrong. This is wrong. We need to find the incorrect statement, and D is the incorrect. Any doubts? Any doubts, students? Any doubts in this particular? Okay, no doubt. Okay, all good. Again, match the following. Match the followings are more. Cristae. Cristae is the infolding. Infolding of the mitochondria. So can you see how many times they have asked about it? Like only few questions asked several times. Cristae is a infolding. Thylakoid, disc-like shaped, no. In flattened membrane, yes, flattened membrane sacs in the stoma of the plastids. Centromere, primary constriction in the chromosome. Secondary constriction form हो रहा था satellite को. Okay. Cisternae. Cisternae is in the Golgi. So which means A3B4. A3B4. B for Bombay is the right answer. C1D2. When the centromere is situated in the middle of two equal arms of the chromosome is referred as. Centromere is in the middle of two equal. This is Q, this is P. P is equal to Q. कहाँ में था? Metacentric में था. So easy as that, it is metacentric. The biosynthesis of ribosomal RNA occurs. rRNA, ribosomal RNA माने rRNA. कहाँ हो रहा था? Nucleolus. Nucleus, where nucleolus is present? Inside the nucleus. The shorter and the longer arm of the submetacentric chromosome are referred to as shorter and longer. Submetacentric यानी, so this is short and another one is long. थोड़ा सा हटके, middle से हटके. So this is P, this is Q. P is less than Q. P is the shorter. P arm shorter and longer. P arm and Q arm respectively. It is B for Bombay. Easy as that, है ना? Easy as that. Which of the following is true for nucleolus? Nucleolus क्या कर रहा था? Nucleolus was actually synthesizing rRNA. Larger nucleoli are present in the dividing cell. नहीं, dividing cell में तो nucleolus disappear होता है ना? It disappears. Nucleolus disappears. Nuclear membrane fragments. So much things. So it disappears. ये नहीं है. It is a membrane bound structure. No, it is not membrane bound. It is actually present inside the nucleus. कोई membrane नहीं है उसका. It takes place in the spindle fiber formation. कौन कर रहा था? Centriole was doing. It is sign of active ribosomal RNA synthesis. Correct. D for Delhi is the correct answer. The chromosome in which centromere are situated close to one end. Close to one end. Which means if this is the end, it is situated. One is long. तो ये हो गया, which is Q is very greater than P. That is acrocentric. So there are chances you marking telocentric. Telocentric, no. Telocentric के लिए terminal आना चाहिए. Terminally. Centromere present terminally. Centromere present terminally is telocentric, as it is centrally situated close to the one end, just close to the one end. This is the end, this is the telomere. This particular part of the chromosome, these parts are called telomere, which is, if this is a centromere, which is situated towards the end, that is acrocentric. Nuclear envelope is derivative of. It is derivative of microtubules, smooth endoplasmic reticulum, rough endoplasmic.
Second, option second.
Yes, P for Bombay. Belbiani rings. Belbiani rings, do you know? This is actually 2015 questions, which now it is not a part of the syllabus, but still, as the question has there, this one Belbiani rings are present in the polytene chromosome. Have you heard of polytene chromosome? Polytene chromosome, actually it is a सलेवरी ग्लैंड इन द डिप्टेरन इंसेक्ट्स होते हैं ना, जैसे ड्रोसोफिला एंड ऑल, एक्चुअली उसके सलेवरी ग्लैंड में ये ऐसे वाला क्रोमोसोम, इट इज़ अ वेरी लार्ज क्रोमोसोम विच इज प्रेजेंट, दैट इज कॉल्ड पॉलीटीन क्रोमोसोम. बाल्बियन रिंग्स यानी क्या होता है पता? इफ दिस इज अ क्रोमोसोम, देन देर विल बी एक्चुअली रिंग लाइक स्ट्रक्चर, ऐसे रिंग जैसे स्ट्रक्चर, लाइक क्लाउडी स्ट्रक्चर ऐसे फॉर्म होता है. ओके, सो हियर एक्चुअली एक्टिवली प्रोटीन सिंथेसिस विल बी टेकिंग प्लेस. प्रोटीन सिंथेसिस एज वेल एज, प्रोटीन सिंथेसिस के लिए क्या चाहिए? आरएनए चाहिए. सो उसके लिए आरएनए एज वेल एज प्रोटीन सिंथेसिस विल बी टेकिंग प्लेस इन द देन सच स्ट्रक्चर. ओके, सो इफ अगर टाइटली पैक्ड हो तो व्हाट, oh my god, Subhash, why did you off it? Okay, fine. Cell, if this is a cell, okay, and this is the nucleus. For example, we'll just take that as a nucleus. The endoplasmic reticulum will be like this, right? The endoplasmic reticulum, then smooth endoplasmic reticulum starts. Now, when I am drawing the Golgi apparatus will be like this. Okay. This is cis phase will be towards the nucleus. This is nucleus. This is nucleus. The cis phase will be towards the nucleus, and here the trans phase will be towards the plasma membrane. It will be towards the plasma membrane. The cis phase and trans phase have other names also. What are the other names for the cis phase? As it is very near to the nucleus, we call it as proximal phase. Proximal phase. Okay. And you can see the convex shape. In the mirror you have learned, right? This is convex, and this is, if this is a shiny surface, this is concave. This is convex and this is concave. As you can see, like this is the convex face, convex. This is like this, right? Like this, shiny surface like this. So this is the concave. As it is far from the nucleus, otherwise it is called distal end. Distal means it is towards the distance, far away, far of the distance. Okay, cis phase, proximal and convex. And another one name it has the forming phase. Formation. So जो भी यहाँ this one endoplasmic reticulum secrete करके देता है ना, it will be going to the Golgi. यहाँ formation होके it will be going under. Formation is taking place, and here it is the maturing phase. Maturing. So here these are the vesicles forming. If it is a enzymes going inside, for example, you take example, I will take it as a lytic enzyme. Okay. If this particular thing is making the lytic enzyme here, and it is giving out this particular vesicle will become the lysosome. This particular enzyme vesicle will become the lysosome. So now it is maturing into something, forming, right? It is maturing into something. So that's why it is called maturing phase. You need to remember these 4 names of the cis phase and this 4 for the trans phase. Trans is towards the plasma membrane, and cis phase is towards the nucleus. Understood? Surabhi? Yes. Yes. Thank you so much. So that is okay. Everybody else, any doubts? No doubt? Okay. So that is the end of the class. Thank you so much, one and all. Thank you so much.